What is cell division? Well, to be frank, it's the division of cells. Duh, but let's go further than that. I mean, what does it really do for us? I mean, it doesn't pay taxes or any sort of rent, so why does it exist in our bodies? It all starts with our own personal cells. Because our bodies are complex and hard to understand, much like the ending of Lost, our bodies have two types of cells, so to speak. Your somatic cells, which are pretty much any type of body cell, and your gametes, which are your sex cells. From these two cells, two processes occur, mitosis and meiosis. Mitosis happens in your somatic cells, while meiosis happens in your gametes. As an end byproduct, mitosis has 2 cells, while meiosis has 4 cells. Mitosis is also more common in multicellular organisms, while meiosis is less common. So while these processes are different in their own ways, they're also very similar as well, comparable to the Crips and Bloods. Before we get into mitosis, we first have to go through interphase, the growth stage of a cell. Interphase is comprised of 3 main stages, G1, S, and G2. In G1, or growth 1, the cell goes through a rapid growth cycle. In S, or S phase, the cell's DNA duplicates itself in order to get ready for division and mitosis. And in G2, or growth 2, all the cell's energy lost in S phase is regained in order to get ready for the division and mitosis. Mitosis, for real this time. Mitosis is split up into 4 main phases: prophase, metaphase, anaphase, and telophase. You can use the acronym PMAS to help you remember this. This is what a cell looks like before mitosis. Notice the duplicated DNA inside the nuclear membrane. You can also see that centrioles are orbiting outside the nuclear membrane in the cytoplasm. In prophase, the centrioles in the cytoplasm move to opposite sides of the cell, or the poles. Then the nuclear membrane fades, releasing all the duplicated DNA, or chromosomes, created in S phase out into the cell as well. In metaphase, the centromeres of the chromosomes line up on the equator. There, the spindle fibers from the centrioles are attached to the centromeres. In anaphase, the middle of the chromosomes, the centromere, are split apart, forcing half of the chromosome to exist on each side of the splitting cell. Just think of it as a divorce, except for both parties get the same amount of stuff and everyone is happy. Lastly, in telophase, the cell is divided in half, which is called cytokinesis, and a nuclear membrane forms in each of the cells, starting off the whole process again. So to put it to example, if some crazy ass homie comes up to you and shanks your ass, the damaged cells from your wound will be replaced with new cells, and all thanks to mitosis. Boom, you're welcome. So once again with mitosis, you have prophase, metaphase, anaphase, and telophase, all in one giant cycle. Meiosis, on the other hand, though similar in nature, has an end product much different than mitosis. Meiosis also is composed of 2 different phases, meiosis 1 and meiosis 2. Prophase 1 is almost the exact same process as prophase, except for the fact that your chromosomes don't just float around anymore, they actually line up side by side, called synapses. This is what these pairs of chromosomes called tetrads all intertwine and break off forming new alleles on each chromosome, making them non-sister chromosomes. So think of all this as a big chromosome slumber party, where things get heated and then everyone eventually exchanges things. Moving along. In metaphase 1, these newly formed sister chromosomes go through the same process as metaphase, except for instead of lining up on the equator as singles, they all pair up as homologous pairs. Once again, anaphase 1 is almost identical to anaphase. The pairs are separated from each other and pulled towards the opposite poles. This is called segregation, and the cell starts to divide. In telophase 1, the cell is now fully divided. The cell now has half the amount of chromosomes as before. For the sake of time, I'm going to leave out meiosis 2, just because meiosis 2 is almost identical to mitosis, except for the fact that really you're just dealing with homologous pairs, and in the end product you have 4 cells instead of 2. Remember before when we were talking about N and 2N? Well, this is called ploidy. Ploidy is the number of set chromosomes inside of a cell. It can be divided into three main categories: haploid, diploid, and polyploid. Haploid, or N, has 23 chromosomes, where diploid, 2N, has 46 chromosomes. Polyploid, or greater than 2N, has more than 46 chromosomes. It's also good to know that haploid N is predominantly in meiosis and diploid 2N is in mitosis. But wait, there's more. Since meiosis occurs with your sex cells, it's good to apply this process to said cells. So, ladies first, we start off with a single oogonium, 2N, who passes through mitosis to create a primary oocyte, 2N. Then, through meiosis 1, this primary oocyte turns to a secondary oocyte, N. And of course, after meiosis 2 occurs, this secondary oocyte turns into an ovum, or an egg, which I'm pretty sure all you ladies are familiar with. They usually bring upon a period of mental and physical changes, so to speak. Unlike the ladies, our process, called spermatogenesis, begins with a spermatogonium, 2N, and divides through mitosis into a spermatocyte, 2N as well. Then, through meiosis 1, it becomes 2 secondary spermatocytes. And then lastly, through meiosis 2, you get 4 spermatids, which eventually mature into sperm. So there you have it, mitosis, meiosis, and everything in between. That's cell division. Now if you excuse me, I'm gonna go take a nap.